Have you ever tasted sprout salad? The image shows the one. You might have heard from your parents that sprouts are a rich source of proteins, right? But do you know that sprouts are germinated seeds of some pulses? The seeds of gram, moong, and other pulses are soaked overnight and then wrapped in a damp cloth to allow germination. These germinated seeds are then garnished and eaten as per taste. Let me ask you a question here. Have you ever wondered how the seeds of these pulses are produced by plants? And what is the process that results in the formation of seeds? You might have seen seeds in some fruits as well. Watermelon fruit has lots of seeds in it, right? So, are seeds found inside the fruits? And if yes, how? Also, how are these fruits that carry seeds in them? Seeds and fruits are the products of events that occur after the process of fertilization in flowering plants. Since these events occurs after fertilization, they are also called the post-fertilization events. Let us discuss these events that form seeds and fruits in the plant. Before discussing the post-fertilization events, let us recall the process of double fertilization. Double fertilization includes a fusion of the haploid male gamete with a haploid egg to form a diploid zygote and a fusion of a diploid central cell with another haploid male gamete to form the triploid primary endosperm nuclei, that is a PEN, right? So, post-fertilization events are the ones that form endosperm from the primary endosperm nucleus, the embryo from the zygote, the seeds from the fertilized ovule, and fruits from the fertilized ovary. Let us discuss these events one by one. The triploid primary endosperm nucleus divides repeatedly by mitotic divisions. It results in the formation of a multiple triploid nuclei, which then form the free nuclear endosperm. The formation of the cell wall then converts the free nuclear endosperm into cellular endosperm, as the endosperm is now composed of triploid cells and not the free nuclei. So, have you seen an endosperm? The answer is yes, as you all surely have enjoyed the coconut water, haven't you? Coconut water is a free nuclear endosperm composed of thousands of free nuclei. You might ask a question here. So, what is an example for a cellular endosperm? The white kernel of the coconut is nothing but a cellular endosperm. Now that you have visualized the endosperm of plants, so can you tell its function? Endosperm tissue serves as a nutritive structure. It is rich in lipids, proteins, and carbohydrates which are used by the growing embryos. So, endosperm nourishes the embryo. This is the reason that the formation of an embryo begins only after a certain amount of endosperm is formed. It ensures the presence of a source of nourishment for the embryo. I have a doubt here. If endosperm is a nutritive tissue used by the embryo, how is it present in coconut? Shouldn't it be consumed by the growing embryo? The endosperm may or may not be completely consumed by a developing embryo. When the endosperm remains persistent in mature seeds, the seeds are called the albuminous seeds. Castor and coconut plants and cereals such as wheat, rice and maize products are examples of albuminous seeds. The persistent endosperm in these seeds is then used up during the process of germination. When the endosperm is completely consumed by the growing embryo and the mature seeds do not have any remaining endosperm, the seeds are called ex-albuminous seeds. Peas, beans, grams, orchids, castor and groundnut plants provides ex-albuminous seeds. I have a doubt here. The primary endosperm nucleus divides mitotically to produce endosperm. But what about the zygote? What types of division occur in the zygote to form the embryo? Also, what happens to the synergids and the antipodals during the post-fertilization development? The antipodals and the synergids degenerate after the double fertilization, leaving only the zygote and the primary endosperm nucleus in the embryo sac. Recall that the egg apparatus is present at the micropylar end of the embryo sac, and therefore the zygote is also present at the micropylar end. The zygote undergoes specific stages of development that together constitute the embryogeny. The early stages of embryogenesis of dicots and monocots are the same. Let us discuss the development of the dicotyledonous embryo in detail. In dicots, the first asymmetric division in the zygote forms a terminal cell and a basal cell, which in turn gives rise to embryo proper and suspensor respectively. The cells of the embryo proper then divides into transverse and longitudinal planes to form a globular stage of embryonic development. The globular stage has several tiers of cells which then establish the three tissue systems, namely protoderm or dermatogen, ground meristem and procambium. With the beginning of the formation of cotyledons, the globular stage of the embryo is transformed into a heart-shaped stage. The heart-shaped stage has shoot meristem and a root meristem developed along the root-shoot axis. The heart-shaped stage is then transformed into a mature embryo. Now, let us check the structural components of a mature dicotyledonous embryo. Cotyledons are the seed leaves of a plant embryo. There are generally two cotyledons. The functions of these leaves is to have stored food such as starch, oils and proteins to facilitate seed germination. Since these food reserves were initially produced as endosperm, the seeds with cotyledons that store food are ex-albuminous with no endosperm at maturity. It nourishes the embryonic plant, that is non-photosynthetic. The embryonal axis includes two main parts depending on its position in relation to cotyledons. The part of the embryonal axis that is present above the level of cotyledons is called epicotyl. The tip of the epicotyl is a plumule or shoot tip. Likewise, the part of the embryonal axis located below the level of cotyledon is called hypocotyl. Hypocotyl has a root tip or radicle present at its lower tip. Unlike the shoot tip, the root tip is covered with a root cap for protection. The radicle and the plumule are called the short embryonic root and the short embryonic shoot respectively. Once a radicle, plumule and the cotyledons are formed, the development of young plants is arrested. It is resumed only when the conditions are favorable for seed germination. You might ask a question here. Do monocotyledonous embryo have the same structure as that of dicotyledonous embryos? The answer is no. A monocotyledonous embryo differs from a dicot embryo for certain structural components. So let us check them one by one. A monocot embryo has a single cotyledon called the scutellum. The scutellum is located towards the lateral end of the embryonal axis. It is a shield-shaped structure and digests and absorbs the endosperm during germination. The embryo also has epiblast. It is a small flap-like structure that represents the rudimentary second cotyledon. Coming to the second point, just like the dicot embryo, the embryonal axis of the monocot embryo also has epicotyl and hypocotyl, which represents the part of the embryonal axis above and below the level of the attachment of scutellum. Coming to the third point, the lower end of the hypocotyl has a radicle and a root cap covered with a protective sheath called coleorhiza. The fourth point, likewise, the young shoot or the epicotyl has a shoot apex and it is also covered with a special sheath of cells called coleoptile. During seed germination, the coleoptile pushes up through the soil and then the leaves and the stem appear from the center of the coleoptile. I have a doubt here. The zygote and the primary endosperm nucleus form the embryo and the endosperm respectively. But what is the fate of ovule, in which double fertilization has occurred and the embryo and the endosperm are formed? The fertilized ovule develops into a seed. So let us discuss the development and the structure of seeds in detail. For that, let us recall the structure of the ovule of an angiosperms. An ovule has a female gametophyte or the embryo sac, inner and outer integuments, micropylar and chalazial ends, and a funicle. Let us correlate these structures with the structural component of a seed. A mature seed has an embryonic plant, food stored in either the endosperm of the cotyledons and a seed coat. We have already discussed the embryo and endosperm that develop in the embryo sac, right? The seed coat is a tough and protective structure derived from the integuments of the ovule. The micropyle of the ovule is present as a small pore in the seed coat to allow the entry of oxygen and water during the seed germination. The germinating seed requires oxygen and water to support its growth, right? When the nucleus is persistent and its remnants are present in the seed, it is called a perispore. The seeds of black pepper and beetroot have perispore. Maturation of seeds include the gradual loss of water content. A mature seed has only 10 to 15% of water content by mass and exhibit a low metabolic rate. Such seeds are said to be dormant and the state is called dormancy. When favorable growth conditions are present, the dormant seeds resume growth and germinate. You might ask a question here. Seeds of flowering plants are contained in fruits, right? So, what are the structures that develops into fruit? Let's find out. Recall that the ovule is present in the ovary of the carpel. The fertilized ovary develops into a fruit. The ovule's transformation into seeds and the ovary into fruit occurs simultaneously. So, as the wall of the integument forms a seed coat, the wall of the ovary forms the wall of the fruit, which is also called the pericarp. So, did you notice the fertilized ovule of an ovary develops into a seed? So, if an ovary has a single ovule, the fruit developed from it would have a single seed. On the other hand, the ovary with two seeds will develop into a fruit with two seeds and so on. We can say that the number of ovules present in an ovary of a carpel determines the number of seeds present in the fruit developed from the ovary. So I have a doubt here. The ovary of the flowers form fruit-carrying seeds which in turn have embryos. But what about the other floral part such as sepals and petals? Sepals, petals and other floral parts degenerate during the formation of fruit from the ovary after fertilization. However, some plants exhibit variations too. The fruits developed from the fertilized ovary are called true fruits. These fruits have only ovarian tissues. On the other hand, the fruits that have one or more non-ovarian tissues are called false fruit, as in the case of apple. Here the fruits develop from the fertilized ovaries, but the thalamus is enlarged to form the bulk of the fruit. So the innermost part of the apple fruit is true fruit as it is developed from the ovary only. Strawberries, pineapples, pears and mulberry are some other examples of false fruits. Let me ask you a question here. We have discussed that fruits and angiosperms develop from a fertilized ovule, right? But can a fruit develop from an unfertilized ovary? And the answer is yes. The fruits developed from the unfertilized ovaries or without fertilization are called the parthenocarpic fruits. Since seeds develop from the fertilized ovule only, the parthenocarpic fruits do not have seeds. You might ask a question here. Is seed formation desirable or not? Let us first discuss the scenario where seedless fruits are desirable. Parthenocarpy produces seedless fruits of pineapple, banana, cucumber, grape, etc. Isn't it desirable to have seedless oranges and seedless watermelon to enjoy these fruits? The treatment of unpollinated flowers with auxin induces the production of fruits without fertilization. You might be thinking, if seedless fruits are preferred, then why do plants invest energy in the production of seeds? Let us discuss the significance of seeds for these plants. The production of seeds is an adaptation to life on land. Seeds have multicellular young plants and have abundant food supply to nourish the plant embryo until it becomes photosynthetic and self-sufficient. The thick and hard multicellular seed coat protects the embryo against unfavorable conditions. Seeds are viable for an extended period with a low rate of metabolism. The dispersal of seeds by the wind and other abiotic agents allows the seed-producing plants to arrive and inhabit a wide range of habitats. The pre-fertilization events, such as the pollination and fertilization in these plants, do not require water, which in turn makes the seed production a better survival strategy. Also, seeds carry the embryo produced by the fusion of male and female gametes. So, the embryo in the seeds produced by sexual reproduction impart genetic variation to the progeny, which in turn are required for evolution. Reduced water content and the metabolic rate of mature dormant seeds allow their storage so that they can be used as propagules to produce the new crop in the next season. However, you should know that the period for which seeds of a particular species are viable after the dispersal varies with species. What do you mean by the viability of fruit? Viability is the ability of the seeds to germinate and produce plants under favorable growth conditions. For example, the seeds of parsley and onion seeds remain viable for 1 or 2 years. The seeds of cabbage remain viable for as long as 4 years. The seeds of Lunipes arcticus obtained during excavation from Arctic tundra germinated after a dormancy of 10,000 years. On the other hand, the seeds of willow are short-lived and remain viable for less than 1 week after dispersal. Now that you have learnt the post-fertilization event that forms the embryo, seeds and fruits. So can you think of a condition where seeds are produced without fertilization? We have already discussed that parthenocarpy produces fruits without fertilization. So can seeds also be produced without fertilization? If yes, what is the process? Also, can a seed have more than one embryo, or it always carries a single embryo? If a seed can have more than one embryo, then how are they produced? We will discuss apomixis and polyembryony in the next video to find the answers to these questions. Now, let us summarize what we have learnt.
Manorama Horizon. Learning made simple.